Good afternoon and welcome to the Harvard-Radcliffe Institute. I'm Immaculata DeVivo and I'm the Melanie Mason Nemec 71 Faculty Co-Director of the Sciences here at Radcliffe. I'm a professor of medicine at the Harvard Medical School and a professor of epidemiology at the Harvard T.H. Chan School of Public Health. And it's my pleasure to welcome you all to our Next in Science program, Next in Alzheimer's Research. We're delighted to have so many of you here with us at Fay House and to be joined by the many hundreds tuning in online. Whether you are across the yard or across the globe, thank you for being part of this conversation. Let me begin by expressing our deep gratitude to the people and institutions who've made today possible. First, I want to thank Harvard-Radcliffe Institute and my colleagues across the Institute who have worked so hard behind the scenes to organize this event, from the planning group to the events and the technical teams to the communications and the facility staff. Programs like this are truly a collective effort. I would also like to recognize and thank our Dean, Tomiko Brown-Nagin, for her leadership and steadfast support of the sciences here at Radcliffe. Her commitment to bringing together scholars, scientists, and practitioners across all disciplines is exciting and allows us to converge on an event like this here today at Radcliffe. We're deeply grateful to Melanie Mason and David W. Nemec Fund for Science and for the generous support of this program. Their investment in early career researchers and in cross-disciplinary science help us spotlight emerging work that has the potential to change how we understand and ultimately how we treat and prevent Alzheimer's disease and related dementias. Today's program addresses a challenge that is both urgent and profoundly human. At the moment, more than 55 million people worldwide are living with dementia. And as the population ages, the number is projected to nearly triple by 2050. Alzheimer's disease is the most common cause, accounting for roughly 2/3 of the dementia cases, and is already a leading cause of disability and death in older adults. Families, clinicians, and communities are feeling the strain in care needs, health system burden, and the emotional toll of these conditions. But at the same time, this is a moment of real possibility. Advances in molecular and cellular neuroscience are clarifying how toxic proteins, inflammation, and vascular changes damage the brain. Genetics and genomics are revealing new loci. These are places in our genes that determine risk, and biological pathways. And research in psychosocial and structural risk factors is showing how education, neighborhood conditions, and lifelong stress shape vulnerability. Alongside these discoveries, clinical trials and preventative studies are testing ways to delay onset and slow progression, expanding the frontiers of what we know about Alzheimer's disease and related dementias, and where, when, and how we might intervene. The goal of the Next in Science series is to highlight rising early career scientists whose work at the cutting edge of these emerging scientific frontiers, and to bring them in conversation with one another and with the broader public. Our speakers today are advancing our understanding of dementia from very different vantage points: epidemiology and social drivers of health, clinical research and prevention, neurobiology and model systems, and the role of lipids, fats, and genetics in neurodegeneration. Together, their work helps us see the complexity of these diseases and the range of opportunities for intervention. Before I introduce the speakers, a quick note of how today's program will unfold. We will begin with 2 talks, followed in each case by a brief conversation with me on stage with the speaker, and after a short break, we will reconvene for 2 more talks. And then a larger group discussion will include audience Q&A. Audience members, as well as audience online, will submit questions at any time during the program, and to use the Slido link provided on the screen behind me, and it's posted in the chat on Zoom. Now, it's my pleasure to introduce our first two speakers, who will open our program by focusing on social drivers and clinical approaches to dementia risk and prevention. Our first speaker is Ganga Bey, an assistant professor in the Department of Epidemiology at the University of North Carolina at Chapel Hill, and an Alzheimer's disease disparities research faculty fellow at the Center for Interdisciplinary Research on Alzheimer's disease disparities at Columbia University. Dr. Bey's work examines how social and structural factors shape aging pathways and risk for Alzheimer's disease and related dementias, with particular attention to disparities across populations. Today, she will speak about characterizing social drivers in aging pathways to Alzheimer's disease and related dementias, helping us think about how the circumstances in which people live, work, and age becomes biologically embedded over the life course. Following Dr. Bey, we will hear from Dr. Jennifer Gatchel, an assistant professor of psychiatry at Massachusetts General Hospital and Harvard Medical School. Dr. Gatchel is a clinician scientist whose research focuses on emerging trends in dementia prevention, including how we can identify and intervene on risk earlier in the aging process. Her talk, Clinical Research, Emerging Trends in Dementia Prevention, will explore how advances in clinical science and care models might allow us not only to treat dementia, but to delay or reduce the onset of dementia. We are tremendously fortunate to have such an outstanding group of early career scientists with us today, and I'm excited to share their work with you. And with that, let's get started. And it is my pleasure to welcome our first speaker, Ganga Bey.
All right. Thank you, Mac. I am really excited today to discuss one of my favorite topics with you all, the ways that I believe the social environment predictably patterns aging and how we can use this knowledge to better the health of everyone. Much of what I will discuss today applies to both Alzheimer's disease and dementias from other causes. So throughout, you will hear me refer to Alzheimer's disease and related dementias, or ADRD. So my talk today details my proposition that we have a lot to learn about ADRD by more closely studying the modifiable ways that human beings hierarchically organize our societies and unequally distribute power. Throughout the talk, I may use several terms with which some of you may be unfamiliar, so things like intersectionality, neuroscience, or identity vitality, but I promise by building out this conceptual diagram to make it make some kind of sense, and or at the very least to make a compelling argument for what social hierarchies might have to do with Alzheimer's disease. So let's briefly talk about disparities. In the United States, ADRD is patterned along clear racial, ethnic, and socioeconomic lines. Some sex or gender disparities exist as well, but they are less consistently observed. And non-Hispanic Black people have twice the risk, and Hispanic persons 1.5 times the risk of developing ADRD as non-Hispanic whites, disparities which persist after accounting for socioeconomic differences between these groups. And similarly, populations of low socioeconomic status has been estimated as having as much as 3 times the risk of ADRD as high SES populations. And this occurs along an SES gradient that also persists after accounting for racial and ethnic differences. And moreover, these patterns persist globally. Across the world, indicators of marginalized or subordinate social status are associated with a greater burden of ADRD. So similar to most other chronic diseases, we know that the contributors to ADRD fall under biology, so chronologic age and genetics, things like APOE, which we'll learn more about from other speakers, and rare mutations for early onset Alzheimer's. Vascular contributors, so your things like hypertension, diabetes, dyslipidemia, obesity, other health conditions like depression and hearing loss, and then of course the usual suspect health behaviors. Okay. So we've added to our conceptual diagram these age-related ADRD risk factors, including a role for genes. And these ADRD risk factors fall into one or more of what I like to refer to as causal buckets, what our genes are, where everything happens, and how our genes function based on where things happen. And so putting aside a future where gene editing is not as impaired by ethical concerns, in population health sciences, we tend to focus on the environment and the gene by environment interaction in identifying where we can intervene to reduce ADRD risk and potentially eliminate disparities because they represent modifiable causes. And given these three buckets of causes, what do we actually know about what explains sociodemographic disparities in ADRD? Modifiable risk factors account for up to 45% of dementia risk at the population level, but explain only 2 to 7% of the difference in dementia risk across low versus high SES populations or across racial and ethnic groups. And this means that the primary drivers of differences in ADRD risk are currently typically unmeasured in population health research. And these factors are often attributed to some black box of gene by environment interaction, that is, some combination of how our genes function in the context of structural determinants, childhood adversity, and chronic stress. To develop innovative solutions for eliminating disparities in preventable disease, it's important for us to tease out this uncertainty. What is this environment that is doing all of these things and causing so much ADRD, but is so poorly understood? What is it exactly that we're referring to, and how does it influence our behavior in our genes? My approach is to look at a very specific aspect of this ubiquitous social environment that I think is really fundamental to all other environmental influences on ADRD risk, and that is the hierarchical organization of social groups in society. And this social hierarchy is characterized by the asymmetrical distribution of both tangible and intangible resources, advantages, and disadvantages across social populations. Now, so far we haven't gotten to anything particularly novel yet. It's an innovative approach in epidemiology, but social hierarchies and the way they function to influence health is also pretty well described in the social science disciplines. And what the social science literature has contributed is an understanding of how to define the social positions in this hierarchy. What they are specifically depends on which country you're in, but sociodemographic characteristics like race, ethnicity, gender, socioeconomic status, these are pretty consistent indicators of a specific social position that an individual occupies across the globe. In the United States, white race, being male, being high SES, indicate dominant social positions, while Black race, female, low SES are indicators of a subordinate or marginalized social position. And what's important to flag is that these distinct and stable social positions are established by a collection of laws, policies, and practices that have been extensively characterized. It's not just a description of any observable difference between groups, but differences in access and resources and power that are established and maintained by state-sanctioned violence. And a primary assertion that I pull from the social science literature is that social hierarchy patterns healthy longevity across social positions, in predictable ways over two primary mechanisms: through imposing disease-causing stress and distributing adaptive resources. In this way, social position is a causal bucket. Through some combination of environment and gene by environment interactions, social position determines what disease conditions will be developed when and by whom. However, things become even more complicated when you account for the fact that few individuals are in a purely dominant or subordinate social position. So what happens when people are in both? What happens when people are in multiply marginalized or multiply dominant social positions? And this is where intersectionality enters, which suggests that these intersecting systems of power create unique exposures at junctions of social position. And the empirical evidence for intersectionality theory has shown us that despite being frequently treated as such, these intersections are not modifiers. They are social locations with distinct sets of causes and pathways. And one potential example is the somewhat inconsistent evidence that the ApoE4 allele, and again, this genetic risk factor for Alzheimer's disease, does not have the same relationship in Black populations as it does in white populations, suggesting some group-specific pathways to Alzheimer's disease. In spite of intersectionality theory becoming more mainstream in population health studies, recently, within group variability, so looking within these groups, is still largely overlooked in and outside of health disparities disciplines. If, however, it is the case that social position is not a moderator, and that these groups located within these distinct intersecting social positions have their own buckets of causes, why not study individuals at these locations as their own distinct groups and look at the within group population attributable risks of ADRD? And this could really help us begin to tease out this black box. So these within groups analyses would allow us to answer different types of questions. What are the collections of causes and mechanisms of ADRD within each of these groups? It allows them to be different. And secondly, why doesn't everyone in that social position develop ADRD at the same time? It allows us to identify sources of resilience and variability that might very well be obscured in pooled analyses. Okay. So we've now added social position to our conceptual diagram. And as we've discussed, these indicators of social position have already been linked to risk factors and genetic predisposition, as well as ADRD itself. Now we begin to dig a bit more into the biological pathways, and the strongest evidence points to chronic stress. It has been argued by universal harm proponents that social hierarchy causes stress for everyone, and it is bad for everyone's health. Here's where we get into the real novelty of my work, where I think I bring the biggest innovation is how I define chronic stress. My novel take on the universal harm theory is that many of the adverse health outcomes specific to dominant groups are unmeasured and are caused by what I characterize as stressors of dominance, and the specific ways that dominant status groups adapt to those stressors. Likewise, there are subjugation-specific stressors and adaptation that corresponds to specific aging, health, and disease trajectories. So stressors of subjugation include stressors that have been established already as a fundamental cause of disease. And these include, but are not limited to, things like interpersonal discrimination, poverty, and physical victimization. And these stressors have been linked by a vast body of literature to the cluster of age-related conditions that increase risk for ADRD. So your metabolic syndromes, cardiovascular diseases, and depression among Black persons, women, and those of low socioeconomic status, among others. On the other hand, stressors of dominance are not really conceptualized necessarily as a function of high social status or really even as health-impacting stressors. And included, for example, are reinforcement of prejudice and bias within one's immediate social environment, high efficacy, high demand occupations, social movements that threaten social position and trigger position-related anxiety, and chronic wealth surplus that enables an access of leisure time and trait boredom that have been strongly associated with antisocial behavior. And drawing on well-supported psychological science, I suggest that the pressures imposed by these stressors of dominance can exacerbate any genetic predisposition and promote a proclivity for social position reinforcement based in risk-taking, control, entitlement, and violence that necessitate a gradual and ongoing rejection of empathy and compassion over the life course. My argument is that these environment and gene by environmental influences cause associated health consequences that manifest in psychological symptoms, which can be understood as personality pathology. Oops, sorry about that. Oops, there we are. And the term personality pathology in psychiatry and clinical psychology broadly encompasses several personality disorders, but can be defined as a pathway of maladaptive personality development over the life course. And several theories have posited person-environment interactions as a determinant. I offered the novel assertion that personality pathology, particularly the so-called dark triad personality traits of Machiavellianism, narcissism, and psychopathy, characterized by reduced empathy and empathy-associated neurological activity, are as much a socially determined health outcome as depression, cardiovascular disease, and ADRD. Importantly, the question that I am putting forth is whether personality pathology is a missed risk factor for ADRD specific to dominant status groups, given that it associates with outcomes known to increase ADRD risk, including heart disease, depression, and social isolation. But then intersectionality theory tells us that we need to get back to those people occupying positions that are characterized by both dominance and subjugation. So your Black women of high SES, Black men or white women of low SES. And I believe that there is also an interactive effect between the types of stressors to which these groups are exposed, such that the effect of one type of stressor on health is exacerbated by exposure to the other. In other words, the more material advantage to which one is exposed, the greater the adverse impact of material disadvantage on health, and vice versa. And I argue that this leads to distinct stress-related health outcomes. So now I have added in the position-specific stressors along this pathway from social hierarchy through social position to known age-related ADRD risk factors, and an additional proposed socially determined risk factor, personality pathology, all the way to ADRD. What is missing is the physiological processes that mediate this stressor exposure and lead to specific health profiles. So based on what we know, I put together this figure that begins to get at that missing piece, aging. The groups located at distinct social junctures are known to have different patterns of ADRD-related health behaviors, risk factors, and health conditions over the life course. In trying to understand how social hierarchy might drive these patterns, I find it useful to focus on this one primary question: why do groups located at these distinct social positions age so differently? And this is where geroscience comes in. So geroscience is an emerging discipline that suggests aging is this gradual, quantifiable process of deterioration across multiple physiological systems, and that distinct diseases or disease states may be better conceptualized as phenotypes of aging that are diversified by environmental exposure. And this makes a lot of sense to me. This framework offers biologically plausible ways that social hierarchy determines these aging pathways to ADRD and why they may be unique at each of these social positions, differences in the pace of biological aging over the life course. So I am arguing that social hierarchy determines how different socially positioned groups age into the specific disease conditions corresponding to the specific combinations of stressors and adaptive resources to which these groups are exposed over the life course. Through this lens, one could argue that trajectories of health and disease, including ADRD, really are just predictable group-specific aging phenotypes. And so what I've tried to do is build the case that biological aging and the geroscience behind it is really the link here between the complex exposures of the social environment and the specific ways that social hierarchy patterns age-related health and disease over the life course, including potentially distinct pathways to ADRD across intersectional social positions. As you can see with this little, I had a little grayed out area up at the top. I do think there is more to the story and another important piece that often gets overlooked in the study of ADRD. And there is a reason why everyone in each of these intersectional social positions don't all have the same outcome and risk for ADRD. And I believe that the impact of position-related stressor exposure, and whether you're talking about stressors of dominance or subjugation, on aging is most profound among those with specific sets of identity characteristics. And to help you understand a little bit better about what I'm talking about here and the nature of resilience within groups located within each of these social positions, I'm going to switch gears for the last few minutes of my talk and focus on what we actually do in my lab. So through the Healthy Aging with Resilient Identities Lab, my team investigates how identity-based factors drive stress-related physiological pathways that lead to chronic diseases that are marked by stark racial and ethnic disparities, so including cardiovascular diseases, endometrial cancer, and ADRD. And in practice, our research focuses on clarifying social structural drivers of distinct aging pathways across social groups, primarily groups defined at the intersections of race, ethnicity, gender, and socioeconomic status. The two research questions that guide this work are patterns of disease observed across these groups actually patterns of aging that are determined by the unique positions these groups occupy in the social structure? And how do individual level, socially determined, modifiable, identity-related characteristics contribute to across and within group variation and manifestations of aging outcomes? So to address these questions, I developed a new interdisciplinary stress and coping framework to pull together all of this dense theory that I just ran through, and to outline the utility for within-group interdisciplinary analyses for understanding population-level aging trajectories and ADRD risk. And this draws very heavily on social psychology to advance population health equity using epidemiologic methods. And this framework, the Identity Vitality Pathology Model, proposes the identity state spectrum, which describes a spectrum from identity pathology to vitality based on 3 dimensions: the degree of endorsement of a self-concept that is inclusive of all living beings, the degree of belief in this intrinsic value of all living beings, and one's propensity toward extending compassion toward all living beings. An identity state is proposed as influential in how individuals appraise and respond to stress, with those endorsing more vitalized identities being less likely to perceive stress in the context of social stressors, and in the instances where stress is perceived, they are more likely to engage in adaptive coping strategies. And greater identity vitality is thus suggested to modulate the relationship between social position-related stressor exposure and aging. I developed and validated a questionnaire, the Identity Vitality Pathology Scale, to measure where on this spectrum an individual falls. So we have, as I said, very quickly run through my argument that social hierarchy is a fundamental cause of the distinct aging trajectories to ADRD through social position. What I have just proposed is a novel source of resilience based in identity that may influence how position-specific stressors diversify biological aging and manifest in specific age-related risk for ADRD for groups located in these distinct social positions. So far, across 5 samples of over 3,000 adults aged 18 to 81, we have found that greater endorsement of a self-concept that is inclusive of all living beings, a stronger belief in the intrinsic value of all living beings, and a higher propensity toward extending compassion toward all living beings, what I call identity vitality, is associated with a lower risk of depression, better self-reported mental and physical health, less social isolation, lower personality pathology, and lower perceived stress. And there remains some important socioeconomic, gender, racial, and ethnic variation to explore further, and a lot more work to validate these findings with increasing methodological rigor, but these initial evidence is exciting and compelling. So I'll end by stating that I aim to make the case that eliminating the excess risk for ADRD, which is attributable to the preventable things that human beings do and do to one another, rests in broadening the scope of biomedical research to more explicitly incorporate the study of social hierarchy. Treating groups located in specific social positions within the social hierarchy as having potential for their own unique cluster of causes and pathways to ADRD may shed light on this black box of gene by environment interactions and enable advances in the science of Alzheimer's disease. And neuroscience may be key here, understanding that the health disparities we are observing may be distinct aging trajectories enables an exciting new avenue for intervening on preventable differences in health outcomes and improving the health from everyone. My work uses an interdisciplinary lens, integrating social psychology, neuroscience, and epidemiology to explore the role of power dynamics in aging. I argue that this interdisciplinary perspective is particularly relevant for complex age-related conditions such as ADRD that are so dependent on life course exposures and may enable progress in eliminating those disparities. Thank you.
Thank you, Ganga. That was fantastic. So the National Cancer Institute many years ago coined this phrase, which is the genes load the gun, the environment pulls the trigger. And so I see the same. Doesn't matter what the chronic disease is. It's the same, right? It's this multifactorial way to get at a disease. I had a really, it's really interesting, your work on identity, the identity states, meaning how someone sees themselves, points to a possible role in reducing the risk of Alzheimer's and related dementias. How might specific identity beliefs, such as feeling connected to others, having a sense of worth, that does depend on social status and being inclined to care for others, buffer or counteract the physiological effects of stress?
So I think that Russ said this stress process, which I like to think of as in 4 parts. We have our stressor exposure. We have the way that we appraise that exposure or that stressor, how we sort of respond to it, and then what we do about that. And so I think the way that our beliefs about who we are and how we're connected with others, where it is acting in that stress process is at the appraisal stage, first and foremost, where when we encounter stressors, whether, as we were discussing earlier, whether it be a tiger or racism, we appraise that as a threat. And I think what we believe about who we are really shapes how we appraise that threat. And that appraisal, in turn, has very significant physiological consequences. Yeah. And so if we have the capacity based on what we believe about our connection to others, we have the capacity to reattribute those experiences. And so I like to think of discrimination, for example, where it is very easy to internalize that someone is targeting you because you indeed are not worthy. But there are other ways to think about that, which is this person is treating me this way because they are suffering in some way. Mm-hmm. And so that, what is called attributional reappraisal, I think it has been shown that it has the very real capacity to minimize the way that we experience that stress. And I think over the life course, if you, especially marginalized groups who are exposed to these life course chronic stressors, really can build up in our bodies over time, and it makes a big difference the way that we enact or the way that we respond to those stressors. And then the second part, where I think these beliefs shape our stress response and the way that it impacts us physiologically, is what we do when we do perceive the stress, right? Mm-hmm. Because in some cases, no matter how much you think yourself connected to someone, it's going to be stressful for you if somebody's mistreating you, or you feel deprived of something that you feel like you have a right to. And what you do about that after the fact matters as much for your health. And so I think what's really exciting about this is that we're seeing that both there in this appraisal stage and the sort of coping stage, that what we believe about who we are has a powerful possibility of influencing how we respond to that stress. And over the life course, I think, really influences our risk for several chronic conditions, including ADRD.
Yeah, so feeling less than has a physiological effect.
Oh, 100%.
Yes. Yeah. We can talk more about that, but I am going to introduce our next speaker. At the very end, we will take questions from the audience and the whole group. Thank you again. Thank Ganga. So it's my pleasure to now introduce Dr. Jennifer Gatchel.
Thank you so much for the introduction and the opportunity to be here today, and to the Radcliffe Institute for all their efforts in putting together this program. I'm incredibly honored. So as Mac mentioned, I'm a geriatric psychiatrist at Mass General Hospital in McLean. And today I'm going to focus my talk and discussion on what is the emerging science behind trends to prevent dementia and promote brain health. So I'm going to try to highlight some of the most exciting things that we're currently doing and kind of what's in the pipeline as well. I want to start with the question I just want to pose to the group. Do you think that the number of steps that you take per day could impact your eventual risk of developing dementia? So I hear a lot of unanimous yeses in the audience. Now, given that, how many of us wear, just out of curiosity, how many of us wear smartwatches or other devices to kind of track step count or other measures of physical activity? And I see almost everyone kind of raising your hand. It's almost harder not to wear these things in the current climate, given the widespread technology that's out there. So I want to start by focusing on recent work from a group of close colleagues at Harvard Medical School and Mass General Hospital, just down the road from here in Boston. And my group of colleagues and collaborators there sought to address this very question using advances in brain imaging technology, and also on drawing on a rich data set that has been compiled in the Harvard Aging Brain Study. So in the Harvard Aging Brain Study, individuals were recruited about almost 15 years ago, and they're continued to be followed annually over time. And they all entered this study relatively with normal physical health, no acute medical problems, and without dementia. And they're followed annually with very detailed phenotyping of their memory and thinking and their functioning, and also brain scans, so the latest technology of how their brain is changing over time, how their behavior is changing, how their memory, thinking, and functioning are changing. So in this work, my colleagues led by Dr. Wendy Yang studied a little less than 300 individuals in this study that ranged from 50 to 90. And at the beginning of the study, these individuals were asked to wear a pedometer, so a pretty low-tech, just waistband pedometer that measured their step count. And they asked them to wear this for 2 weeks, and they averaged the step count. And then they followed them over time with kind of annual memory and thinking tests and brain scans. And in short, they found that the more steps that folks did, the better that they did on memory, thinking, and functioning tests over time. But a key takeaway here is in this visualization that they show here. And so first, to walk you through this, they divided the individuals into red and blue. So red were the individuals that had evidence of biological risk of Alzheimer's disease in their brains, even though they did not have dementia. So they were able to visualize that on brain scans. And blue were the individuals at relative low risk, so no evidence of pathology in their brain. And again, all are non-demented, at the beginning of the study. And they actually found, and then here on the x-axis, the horizontal, they divided activity in increments between 3,000 steps to 5,000 to 7,000 and greater than 7,000. And to put this into perspective, the difference in about 2,000 steps is about like 20 minutes of walking or an equivalent degree of physical activity over time. And so also on the graph, lower values indicate better memory and functioning. So the key takeaway here was that although everyone did better with more physical activity, with even incremental increases of like 20 minutes of walking per day, the high-risk group in red began to look like the low-risk group, to the extent that in the most active individuals, they almost had identical performances over time on memory and function tests. So this was quite interesting. And again, it highlights that the modifiable risk factor of physical activity, and also a message that, what do we mean by physical activity? That even small incremental changes can actually have biological effects on our brains. So it doesn't have to be a degree of like all or nothing. So that was a very exciting finding. So physical activity is one of the pillars of brain health. And we've probably heard about brain health factors, what's healthy for our bodies, what's good for physical health and our heart, what's good for our brain. And so these pillars of brain health, in just kind of general terms, they map onto the scientific modifiable risk factors that Dr. Ganga nicely presented in terms of if we could together address modifiable risk factors, we could prevent up to almost 40% of age-related dementias. So I told you a little bit about physical activity, and today I want to, in the first part of the talk, pull back the curtain on a few additional pillars of brain health, such as brain healthy diet, mental health, which we all know is very important, and social engagement. And of course, these are all related to each other and also to medical conditions. So I told you a little about physical activity, and I'm going to start with telling you a little about mental health. And of course, these things are related. And one of the ways that we were able to not address these questions in a novel way in the Harvard Aging Brain Study was through the advancement of the ways that we can image our brains. So through time, from the 1800s when we used X-rays to the 2000s when PET scans were developed, we can not only measure in living individuals structure of the skull and eventually the brain tissues, the unique areas of the brain, but with PET scans on the far right, we can actually measure in a living individual activity, like where certain proteins are, where they're distributed and the amount that are there in living individuals. And this is actually just the tip of the iceberg. So PET scans, we can then not only measure activity, so higher activities in red compared to blue, but then we can also introduce specific antibodies that are linked to tracers. So can we measure where is amyloid in the brain or where is tau in the brain or other molecules? And we've even seeking to see if blood tests can correlate with these findings. And if we can measure even more molecules and combine these modalities while individuals are even performing tasks of memory and function. Okay, I'm not going to get too excited and talk all day about PET imaging, although I definitely could do that. But I'll show you one more picture here, and this is just 3 individuals where they all underwent, in the first column, amyloid and tau PET scans. So these tracers came up in the early 2000s, and already a great degree of advancement of the specificity of tracers that are available. They're binding specifically these proteins, and also our ability to measure other measures of pathology that we know are important, like we know it's not amyloid and tau alone. So in the first column, this is an individual who's an older individual who doesn't have dementia, is functioning normally, and we can see relatively no tau and some trace amounts of amyloid there in the medial temporal lobe. The middle column is an individual that we would call clinically in the preclinical stage. So not really showing much signs of change, maybe reporting some subjective memory concerns, but yet there's a degree of amyloid in the cortex and also some early signs of tau deposition. And finally, the right column, this is an individual who would meet the criteria for dementia. And we would see even more widespread amyloid, and now we see tau spreading in a widespread fashion as well. So we're going to hear a little bit more about amyloid and tau and that cascade in later talks, but I just wanted to show you this technology, which we used in the physical activity study. And we also use this in a study that I began in the Harvard Aging Brain Study to look at the relationship between depression and whether or not this accelerates dementia. So we all know from being at this session that dementia is incredibly, of all types, Alzheimer's disease and related types are incredibly prevalent. By dementia, we mean both an alteration in memory and thinking. So it doesn't have to be memory, could be another type of thinking that's affected, and it's severe enough to affect one's functioning. So again, dementia is increasing prevalence with the aging of our population. And also depression in mid to late life is also incredibly common. And this can either be a manifestation of aging with depression or having worse depression as one ages. So given these incredibly important public health factors and my own kind of seeing this commonly in my practice as a psychiatrist, I wanted to ask whether or not depression was linked to potential worse cognition, whether or not it interacted with pathology. And so also in the Harvard Aging Brain Study, in a group of about 300 as well, we looked at depression on the x-axis and then cognition on the y-axis or vertical axis. So we see that increasing depression was associated with poorer performance on cognitive tests over time. But this only occurred based on the degree of amyloid that individuals had in their brain. So this was interesting to us because no one in this study had dementia when they entered the study, and also no one had significant depression. But changes in those behaviors over time or emergence of symptoms led to worse performance. But this also was modified by a biological factor. So this is interesting to us because we think about, can we treat depression? Can we target depression if we catch it emerging to shift the curve of someone in this really downward trajectory, to shift it further up? So that's indeed what we're trying to ask in the Department of Psychiatry to clinical trials and related studies. On the flip side of this, I started a related study, the Mood and Memory and Aging study, where I specifically recruited individuals who have aged with depression and kind of asked, how does that lifelong depression confer risks for dementia in late life? Because certainly not everyone with depression is going to develop dementia, but what can we identify specific individuals who are at highest risk, and can we deliver better treatments to them to prevent dementia? And so as we know, depression and mental health, and we've heard very nicely from Dr. Bey, again, this relationship between social determinants, social engagement, and mental health. And so also one of the advances that we made together with Hiroko Dodge, a colleague now at Mass General, is going to try to break down what could we do to harness social engagement in their broadest sense. So we all know there's many, like in terms of psychological factors that we've really elegantly heard about in a really elegant way. How else can we intervene on this factor of social engagement? What does it take? And so in this study, it was actually, it began during COVID. And so Dr. Dodge and colleagues followed a group of individuals who had early stages of cognitive impairment. And they were living at home. They were socially isolated, so that's not too surprising during COVID. They had minimal depression. They actually introduced a internet-based social engagement platform for the individuals where they chatted virtually with the same community volunteer over a period of 12 months. And so this was like about weekly chats. It was always about just like common everyday topics. It wasn't anything typically like profound. It wasn't psychotherapy. It was just kind of chatting with kind of the same individual over time about current events. And they actually found that those individuals who had that intervention or the iConnect intervention compared to just receiving kind of like handouts or occasional calls from study volunteers actually did better in their overall memory performance over time. So that was exciting because it again breaks down what do we mean by social connection. It's very complex. There are multiple ways that we can target it through a lifespan approach. But this shows that we can intervene or even making a change, such as ongoing social interaction or coming to a talk like this or continuing to refer back or meet up with a colleague on a regular basis. That can have an impact even at early stages of dementia. So we're seeking to replicate this in individuals that have depression and even higher risk group and see if this type of social engagement can help. Now, the last pillar I want to shed a light on is diet, and I know everyone's always very interested about diet and what is the diet or supplement that I can take that would be most protective. And so I can tell you there's no one magic bullet. I wish that there was, that we can say if you followed this diet or took this supplement, you will do better in terms of brain health and preventing dementia. But the evidence seems to be converging on 3 diets: that the DASH diet, which is targeting high blood pressure, so trying to reduce blood pressure, decrease excessive salt intake; the Mediterranean diet, which is composed largely of colorful legumes, fish, olive oil, the food shown at the right, and also the integration of these diets, so the Mediterranean and DASH intervention for neurodegenerative delay. I would say the evidence is the strongest for these 3 diets. And in a recent study from Harvard, actually at the same time that the physical activity was coming out, this group from the School of Public Health actually found that there was an interaction between genetic risk, so ApoE4 gene, and how protective dietary modifications were. So this is, again, telling us that it doesn't have to be an all or none change. There does seem to be an interaction between degree of risk and what can confer protection, indicating at a personalized, maybe a prevention pathway in the future. So just briefly, I just wanted to put up here, folks often ask, what does this mean? So generally, don't want to make this overly complex. Generally, you want to increase these foods and nutrients on the left and decrease on the right. And again, it doesn't have to be an all or none, changing completely from the left to the right, but like one specific step could be maybe swapping out one of these servings on the left for one on the right in a given week. And again, our evidence is suggesting that even the small incremental changes could have an impact. Now, in the final series of the talk, I want to go over what is kind of emerging in terms of what is coming in the pipeline, what is reaching the clinic already in terms of strategies to prevent dementia and to slow its progression once it takes root. And so I briefly want to touch upon disease-modifying agents, nutrients and supplements, and novel agents and combinations of existing agents. This is just a snapshot of emerging therapies in this area. So such as antibody therapies, we now have the first FDA-approved therapies that combine amyloid and tau in the brain, and we can deliver these via infusion. But beyond that, also studying at how can we target other molecules, how can we utilize mineral homeostasis, nutrient homeostasis to slow or prevent dementia? And also, how can we modify brain activity in living individuals through neuromodulation? So through TMS, or transcranial magnetic stimulation, which is a treatment that we deliver commonly for depression, where interestingly, you can just walk in, get the treatment, walk right out, no negative side effects. And there's also ways that this type of brain stimulation could be done remotely in a person's home, so decreasing the barrier of having to come into a clinic. And couple that with other brain training to have a beneficial effect. So again, lots of things are on the horizon here. And then my work, in addition to focusing on depression and behavioral factors, it really gets at trying to create personalized risk profiles for individuals through brain imaging studies, through characterizing behaviors and mood on smartphones, through even blood tests, again, that can measure additional prevention factors. And the goal is to identify, again, for a given individual, what would be the right intervention at the right time, and also what would be the right dose. So that's what we're ultimately aiming at with this approach. And so we're excited. This is a really great program of precision psychiatry at Mass General. So we're excited to kind of work at this interface between dementia prevention and precision psychiatry. And further, I'm just going to close here with saying that our vision for dementia prevention and brain health involves not only, I've focused a lot on the biology because there's some very exciting advances that I wanted to share and some implementable strategies that we can utilize. But also, as Dr. Ganga presented, and as we all know, this goes way beyond biology. This is an incredibly complex illness because we're talking about individuals with different developmental histories and backgrounds and experiences that they've had. And also their family units, so their care partners, and the stress that this can confer to family members, which we know it can be a significant risk factor to care partners. And how does that then affect patients? And so I just wanted to highlight that we're trying to also advance clinical research through not only biological studies, but also through system-based studies. So in Caring for America's Aging, this is an example of a novel study. We're trying to use transportation solutions, so rideshare services, to get home health aids to people with impairment who are at home, because that's one of the biggest barriers to someone's getting a home care, is someone to be able to get there, especially in a city like Boston or where that's very congested. And so we also want to study ways that we can deliver this care in the clinic in an integrated way. So instead of someone going into one place for a psychiatrist and neurologist, can we bring all of this together into one embrace? And so that is our vision, ultimate vision in terms of clinical innovation at Mass General Brigham. So our goal is prevention of illness and increased quality of life for patients and their care partners. And the reason why this is so important to us, it starts and it really ends with the patient, for me and for us in this field. So this is a picture of Rose Ganucci, and she's an Italian immigrant who is really the heart of her family. And this is her granddaughter there, which happens to be me. And so she was the heart of our family. And really at this time, she began to develop early signs of dementia and also Parkinson's disease. And no one in our family had a medical background. So it was incredibly, as many of us know, it's incredibly frightening to be like, what's happening? What's going on? And so just the compassion of having a doctor who really held her hand and connected with her made a huge difference, beyond anything related to medical science. And she did benefit from medical intervention. So she took interventions for Parkinson's disease, which definitely extended her quality of life. But she eventually passed away from dementia. And this is when I'm just entering college, and Nada Rose kind of towards the end of her life. So my work is really dedicated to this. So it's both kind of a professional journey. Wish I could put patients' families up here. It's also very, very salient, kind of hearing their stories, and also a personal journey as well, where we can appreciate kind of the impact on psychological factors, families, economic factors as well. So it looks like I'm nearing the end of time. I've talked about a lot today in terms of resources. Just as I mentioned, just by being here, this is a protective factor for your brain. And also, there are many publications through the Harvard Health System. You may be subscribed to some of those. At Mass General, we put together one, the Mind, Mood, and Memory kind of newsletter, and then we can compile the special issue on boosting your brain power. But this is just one of many kind of in the system, in the Harvard system, that kind of compiles this information. And we try to break it down into SMART goals, because it's not only important to identify what can we do, but also how do you implement that change? So you can tell me, I have a better diet, but how am I actually going to do that? Or what are the barriers to that? So SMART goals, setting a specific, measurable, achievable, and relative time-bound goal. So I'd like to thank NJH Department of Psychiatry, Harvard Aging Brain Study, all of our wonderful study participants, patients and families, my family, and the Radcliffe Institute. And if you remember nothing else from today, just the key takeaway is you don't have to run a marathon or the equivalent to prevent dementia or make a mark on brain health. Every step counts.
Thank you, Jennifer. That was great. That's a lovely picture of you and Nana.
Oh, thank you.
Yeah. We don't have much time right now, given that we're going to take a break, but I did have a question and want to tap into your expertise as a physician. Can you tell us more about new medications to treat Alzheimer's and the new kind of blood test that you were suggesting?
Yeah, so basically we now have two of the first FDA-approved medications, lecanemab and donanemab, that are approved for early stage Alzheimer's disease dementia. And what's really important is these are the first medications that we can administer. They can actually enter the brain and the nervous system, bind amyloid plaques, and remove them over time. So that kind of led to their basis of FDA approval. Now, they're not curative for Alzheimer's disease, interestingly. So the research trials that led to their approval show that they kind of slowed progression or slowed decline modestly when it was present. But still, that degree of slowing and kind of delaying and increasing time for meaningful activities and quality of life is important to many people.
Of course.
So those are available clinically, and they do come with a certain degree of risk, but but they do offer hope in terms of we're now able to remove these proteins, and what's next in terms of using combination treatments or even personalized approaches. And we haven't really, it's too early to know what happens over time, so we're kind of gaining that information right now. In terms of blood tests, the canonical way they would diagnose Alzheimer's disease, if you do a PET scan and see amyloid, we now have a blood marker, phosphorylated measure of tau, and we'll hear more about tau protein, but these are kind of coupled proteins in Alzheimer's disease, and it's thought that amyloid is kind of the first actor, but then tau comes along and is modified and kind of does more of some of the dirty work. And so this p-tau marker seems to correlate to a degree of not only amyloid positivity, but maybe a little bit more progression. So it can also be a way to test, is the dementia or the symptoms related to Alzheimer's type dementia or another type?
Well, that's great. We're at time, but we will have a lot more questions at the Q&A. So we're going to take a short break right now for people in person as well as online. We'll be back promptly at 3:10. And thank you. So far, so great.